welcome to this session. Welcome to the speakers and moderators, and welcome to all you in the audience. I'm Anne Cambon-Thompson. I'm a medical doctor, French, and a researcher in immunogenetics and health ethics, and involved in, since about 20 years, in activities related to ethics in genetics. Among other things, I am a member of the European Group on Ethics of Science and New Technologies, so-called EGE, and I was the champion of the previous ESOF in Toulouse in 2018. On behalf of the brilliant panel you're going to listen to, I thank a lot the program committee and the organizers of ESOF 2020 to have chosen our topic in that session to be debated here, although virtually. And I thank the speakers and moderators to have accepted to share their views and their enthusiasm, and you all in the audience today. So the topic, with the rapid dissemination of genome editing across countries and scientific fields since the 2012 publication of CRISPR-Cas9 technology, societal and ethical debates have developed. This technology and other newer ones, more precise, they are more precise, easier to implement, and cheaper than previous ones. And therefore, they disseminate very quickly among research laboratories and many domains of application. As they may be applied to many fields and species, human cells, microorganisms, animals, plants, several applications re-question values, establish frameworks or regulations, and I mentioned some of them. Human genome modification in a way transmissible to future generations. Blurred frontiers between therapy and enhancement. Humanization of animal models, eradication of species that are vectors of diseases through gene drive, qualification of organisms with an edited genome as a genome modified organism or not. It is thus important to discuss how to regulate the application of genome editing in humans, but also in plants in relation with the use of genetically modified organisms, in animals regarding animal welfare and protection of the environment. A number of bodies, both scientific and ethics, have issued positions on such aspects at national, European, or even international level. Some events actually fostered debates and positions, such as in November 2018, the revelation that a Chinese researcher did modify at embryo level the genome of twin girls for a gene involved in HIV cell penetration. The twins were subsequently born with a certain level of edited genome. So lots of debate about that. The European Group on Ethics of Science and New Technology that I was mentioning before provides the European Commission with independent advice on ethical aspects of science and new technologies in relation with EU legislation or policies. The EGE is an independent advisory board of this European Commission, and such a body exists since 1991 and is regularly renewed, comprising presently 15 members. The EGE has issued a short statement on genome editing already in 2016, and is currently working on genome editing, ethical and regulatory dimensions in different contexts and species, focusing on the European dimensions. And this opinion is expected in the fall of 2020, and its preparation has motivated a public roundtable in autumn 2019. Thus, it is not possible to present this opinion final content today. And as a matter of fact, this opinion has been delayed due in particular to the COVID-19 pandemic that changed the way we were working. But there are many reports and opinions on the topics. And many address the course of genome editing of the human genome, including and focusing on the question of the legitimacy to consider human genome edition in a way transmissible to future generations, which is for the moment a red line for most. While the application of this technology to therapy and somatic cells is considered less problematic and able to provide new therapeutic waves. Such statements and opinions that continue to appear regularly get different echoes among general and scientific media, policymakers, the public, and different communities. Such opinions are playing a specific role in reshaping the science-society dialogue. And our aim in this session is to examine from different perspectives how genome editing is a specific example of a domain with multiple facets, not only scientifically, but in its relation to policy, to societal dialogue, to regulatory frameworks, and to decision-making in uncertainty. And we do have an excellent panel for that from across the world, with a tandem of lively moderators, as indicated on this slide. Each of them will give some more details on their own presentation while starting their talk. And we have chosen to focus more specifically in their short talks on the non-human genome domains. But of course, in the discussion, all domains, including human genome edition, can be addressed. So I will now pass the word to one of our moderators, and the moderators will also present themselves, sorry. And so I'm going to conclude and to pass the word to Marion Abécassis to continue the session. And I wish you all a lively and interesting discussion. Thank you.
Hello, everyone. I'm delighted to be meeting you today in those strange circumstances. So thank you very much for your introduction, Anne. So contrary to Anne, I am not a scientific researcher, but I'm a lawyer. I'm Marion Abécassis, and I've been fortunate enough to be following the works of the French Inserm Ethics Committee for over 5 years now. And I'm a proud founding member of the association ARIGE, which means the Association for Responsible Research and Use of Genome Editing Technology, that aims notably at disseminating reliable information on this technology, and at fostering debates, including with the lay public. So discussions today come at the right time, because just a few minutes ago, the US National Academy of Science and Medicines and the Royal Society in the UK released a long-awaited consensus study report on heritable human genome editing, which is not the first, but will certainly not be the last report on the subject, just as Anne pointed out in her presentation. And those institutions gathering their thoughts and analyses and providing policy recommendations, I could not think of a better transition to turn to Eric Mélin to present to us the role of ethics and evidence in gene editing policy development. Thank you.
Merci, Marianne, and thank you very much, Anne, for extending the invitation to this group, and welcome to my fellow panel members and those of you either in Trieste or watching globally. I'm Eric Neslin. I'm here wearing a couple of hats. I'm a bioethics and policy scholar and administrator, having watched and participated in many of the debates on topics in science and technology for many decades. My current role as president and CEO of the Council of Canadian Academies is not one that I will be wearing fully today, although the organization, very much like the US National Academies or the Royal Society in the UK, undertakes assessments of evidence on behalf of important policy topics. So I'm going to be sharing some reflections today on how I think the role that ethics and policy can inform and has in fact informed debates on this topic. By way of background, and perhaps not spending more time than is needed, I think Anne summarized quite well how the topic of gene editing captured our attention, probably at least since the April 2015 paper in Protein and Cell, reporting on the results of a CRISPR-Cas9 gene editing technology to remove a mutant globin gene from a non-viable embryo. That attracted world attention. The results, of course, were quite promising, to use a much used word, that the technology might be adopted for widespread use. The capacity to cut or edit, and we'll hear more about the use of those verbs later, certainly caused great interest among scientists, ethicists, funding bodies, advocacy organizations, NGOs, industry, the media, politicians, and of course the public, who all began staking out various positions ranging from the most restrictive to the most permissive. And in the past 5 years alone, we have seen these positions evolving, perhaps maturing, and in other cases solidifying in extremely important ways around a known set of topics familiar to those of us who've been engaged in or watching genomic bioethics for human, animal, agricultural, and other uses for years. So I want to just highlight 3 or 4 areas where I think ethics and evidence may play a role and are playing a role in this debate. First, with respect to the role of ethics and evidence. Ideally, and I say that with a hopeful word, good policy is best developed on the basis of good evidence, or at least sufficiently robust information about what is known, what isn't known, and what the implications of that gap are. The shorthand, evidence-based policy, very much like evidence-based medicine, has long ago been, if not debunked, certainly replaced by a much more pragmatic phrase, evidence-informed policy, something where we understand that we should know something about the topic before we start regulating it. Similarly, it's also expected that good policy is best developed on the basis of a sound and supported ethics foundation, whether this is on foundational moral principles, political values or commitments, or as in some cases, and gene editing is one of them, on a more specific set of principles and commitments. Now, we use the word ideally, or I say ideally, because this ideal is rarely met. Sufficiently robust evidence, well-supported ethics foundation, especially in a fast-moving area of science. And sometimes policymakers have to settle on, or might even prefer, a minimal set of data, a minimal set of facts, and a presumed set of values. So I say that just as a background comment. Let me just highlight a few obvious things about the gene editing story. First of all, there's a lot of information, a lot of evidence, a lot of knowledge arising in a much more open and democratized environment of access to information. I hate to say this, but I did a Google search of the term gene editing, and there are 239 million hits by typing in into Google the phrase gene editing. If you type in gene editing professional guidelines, you get 98 million. You get the idea. Type in a term in a search engine and lots of things come out. There is very little discrimination in that open environment between the peer-reviewed literature, the gray literature, blogs, opinion editorials, reports, patent applications, and the media. So some of the considerations that flow from this information tsunami, of course, is that there's an uncertain epistemic hierarchy. What counts as really good information that should be relied upon, and what is just sort of passing anecdote? And how does one discriminate between those? A second consideration is that there are many, many organizations and groups weighing in, some invited, some just entering the room of public policy on their own accord. Anne mentioned some of them. Of course, there are governments themselves and the various orders of government, national, regional, provincial, state, even local city and municipal governments are saying things. But so too are the learned societies of particular countries, national bioethics commissions, regional organizations, professional and other science groups. Even NGOs and larger organizations like the WHO are actively engaged in this topic. Now, we've just learned about the National Academy of Medicine, National Academy of Science Royal Society report. I've not read it. I was preparing for this presentation, but we can tell you that there are a few things that I think are true. One is that this is simply the next one in a long chronology of reports that have come out. We actually have to read things in order, and I honestly worry about people, the phrase is cherry picking, taking a report that they read and using that as the latest evidence. It's quite hard to do this in such a fast-moving environment. There is no World Science Court or World Ethics Court to adjudicate between these different documents, which one is better, which one is worse. A third area is that there are a number of different policy instruments that might flow from those many reports. And of course, the list is quite long. There are declarations and treaties and conventions. There are trade agreements. There is hard and soft law. There is regulation, professional guidelines, standard operating procedures, IP regimes, and the like. No two of them are the same. They flow from different policy orientations. They are designed for different uses. So one can't just pick, the Oviedo Convention and say that's the document that we will use to guide future policy, when we're going to hear from the European Group on Ethics, who might have a very different view. So there are different interests, different governance tools. There are different levels of specificity of those documents, different types of agreement that occur. And I suppose finally, I would say that I alluded to the ethics part at the very beginning of my remarks. This list of principles or values or commitments is as diverse as the policy documents and instruments and groups that construct them. In fact, there is no shortage of policy documents, and there's no shortage of ethical principles, values, and commitments. In fact, if anything, we may have more than we need, so many more than we need that we don't know how to adjudicate between them, how to rank order or lexically order them. At a level of foundation or generality, it's very hard to disagree with scientific freedom or protection of human or animal welfare. It's at that level of specificity where the detail goes. So let me summarize maybe a couple of final points before I end. I think policy is both a goal and it is a tool. And we have to understand when we are using it for a particular purpose. Secondly, while gene editing is a particularly salient point today, it's not the first time society regulate them. We should not forget that there is a history before today on issues around the design of new organisms or the like. And lastly, we might find some comfort in knowing the international community is focusing on these issues, but we should not forget that at some important level, there is a local concern. The very region, the very city, the very community, which might be most affected by, say, a release of a genetically modified organism or something else, will always capture public attention and not limited to what's happening around the globe. Thank you very much.
Thank you, Eric, for this very clear and interesting presentation. So indeed, reports and policies or lack of policies on genome editing are rooted in a specific context. But when we take the context of humanity, we know that men have a long history of modifying their environment, not only physically, but going to the very core of the DNA of plants surrounding them, whether men were aware of it or not. So I will now turn to Julian Kinderlehrer, moving from Canada to all the way to the UK or South Africa, because our speakers have not only many hats in terms of functions and titles, but also in terms of countries they live in or come from. And Julian will speak to us regarding the use of genome editing in plants. And because he also used to be a professor in IP law, and I'm not the only jurist in this panel, maybe he will a little bit talk about intellectual property considerations regarding gene editing technology. So I leave the floor to Julian.
Thank you, Marianne. I sort of introduced myself. I'm a former chair of the European Group on Ethics, and I've been involved in the regulation of genetic modification since 1979, an enormously long time. The talk I want to give today specifically addresses the issues relating to the editing of the genomes of plants. The use of these technologies in agriculture is already ongoing and has or will replace almost all technologies in agriculture for the modification of plant genomes. Previous gene modification technologies caused significant controversies, particularly in Europe. We didn't see that concern in North America. And growing plants containing modified genomes has largely been banned in the European Union. Many, especially plant breeders and most scientists working in the field, argue that these technologies are fundamentally different, the new ones, the gene editing ones, and therefore the precautionary concerns that Europe had are no longer relevant for the vast majority of modifications that might be implemented for agriculture. So my first question that I wanted to ask you and ask myself, is genome editing different from other techniques for producing new varieties of plants which are used for food, feed, fiber, and so on? Selection has been used for centuries to produce domesticated plants that are extremely different from their wild precursors. In the 20th century, deliberately introduced mutations using chemicals or radiation or genetic modification techniques were used to make new varieties. The ability to regenerate a complete plant from a single cell in many instances made all of these techniques useful. But the changes were random, and selection and backcrossing techniques had to be utilized over a period of time to choose and stabilize desired characteristics in the appropriate plant. Genome editing, on the other hand, is increasingly able to precisely locate changes in the genome, and these changes range from the modification of a single base, say to change a protein that is found in one plant to that which is found in another, all the way to major additions or deletions in the sequence of the genome of the plant. Plants can be very varied. The baobab tree has just been shown to have an enormous number of chromosomes. It was over nearly 200 compared to our 22. Where a change in the genome involves many new protein and enzyme genes, or where the position of changes is random, it's clear that a risk assessment must be very detailed and deliberate if we follow the precautionary principle, in order to avoid or minimize the risk of serious or irreversible harm to either the environment or to human health. And that has been the position of the European Union. Where the changes in a position in the genome, which has been well characterized, and the change is one which it renders that portion of the genome similar or even identical to that found in similar organisms, a proportionality requirement should indicate a light-handed approach. And that's the view of most of the scientists that have been involved in this area in Europe. In June 2018, the Court of Justice of the European Union decided that organisms obtained by new methods of mutagenesis are GMOs. They are genetically modified organisms, and are therefore subject to the obligations which are required through the various GMO directives. This has been particularly implemented in France, as our two moderators are French. I comment there. They excluded from this decision organisms obtained by mutagenesis, which have conventionally been used in a number of applications and have a long safety record, because they were interpreting the actual law that existed in Europe. The court understood that mutagenesis permits the modification of the genome without the insertion of foreign DNA, unlike transgenesis. But the court decided that organisms obtained by mutagenesis are GMOs within the meaning of the directives, insofar as the techniques and methods of mutagenesis alter the genetic material of an organism in a way that does not occur naturally. It follows from their decision that gene editing will result in organisms that fall within the requirements of the directive and therefore need to be assessed, and a risk assessment has to be performed. It's what that risk assessment is that will need to be taken into account. What therefore constitutes safe? Because that becomes a major issue, possibly the main issue. Is a new variety to be tested on the basis of as safe as that currently on the market? Does the new variety have to be safer than that currently used? Should the whole system, including chemicals used, land used, and protection or otherwise of agricultural diversity, be taken into account in deciding on safety? Should the requirement for a risk assessment be based on an examination of the potential benefits and costs of action or lack of action, including where appropriate and feasible, an economic cost-benefit analysis be part of the risk assessment that we have to do? It is likely that modifications introduced through conventional breeding techniques, occurring naturally or through gene editing, may not be able to be distinguished one from another. We don't necessarily introduce some form of marker to make it easy to differentiate between that which has been gene edited and that which hasn't, particularly if we work in different jurisdictions. Can we introduce a system for traceability? How do we address that? Would such a traceability criterion work, given that the products are grown throughout the world? And I can't see North America or China agreeing to a new requirement for traceability. So if Europe asked for a traceability requirement, it would actually mean that we wouldn't be able to import anything from other countries which don't have it, because there's no way to differentiate between the gene-edited organism and any other. There is a need to ensure food security. We need to provide renewable resources for fuel, feed, and fiber, and ensure the retention of biodiversity and protect our environment. Gene editing technologies could, with appropriate and absolutely proportionate control, enhance our ability to achieve these goals. There is one other problem that is appearing very much on the horizon and which Marion has started to address. And that is that many of the changes we are making are protected by patents. One, that means that characteristics which a farmer might want, which come from different manufacturers, would not be able to be mixed. If something's been produced by Bayer and something by Limegren, how do we assure that both those new characteristics are in a plant which a farmer wants to grow? That's one problem. But a second is the enormous cost associated, one, with going through the risk assessment, and two, the implications of paying for the patented variety. And both of those impact on the price of the goods and the ability of small producers to produce something which might be used in the environment. So both of those impact very seriously on our ability to use these new technologies. And I hope I'll get some questions on that. With that, I think I've tried to address most of the issues. There's justice issues. There's all sorts of issues that arise from the use of gene-edited plants. Thank you.
Thank you very much, Julian. And I must say, for the moment, the speakers are exactly on time. So my role is very minimal. So thank you, Julian. So we see indeed, and it makes me remember the first thing that I've been told when I started studying law was that law is a fiction. So whatever decisions the European Court of Justice takes regarding what is a GMO or not might not be quite the same as the scientific reality underlying it. And I don't know if you have seen these Netflix shows called Unnatural Selection, but it also stresses out the issues of economic and justice, economic problems and justice value not being enforced enough when you have this conflict of norms. And in fact, if you look at this TV show, it starts with a dog breeder that turns into a biohacker using gene editing on dogs. So actually, we will move a bit from the use of gene editing on plants to go on other kinds of living beings with Perret, who will address, will maybe complete a little bit what Julian said from a more scientific perspective, and touch base with the use of genome editing in farm animals. So if you are ready, Perret, the floor is yours.
Yes, thank you very much. It's a pity not to be able to meet in wonderful Trieste, but I mean, it is what it is. So I am a molecular biologist. I am in Barcelona, closed in my room. And I've been a member of the European Group of Ethics for 13 years. So we have had time to discuss some of the issues that we are discussing. In fact, I am going to build my presentation on 3 opinions. These 3 opinions are available to the public. I encourage people to look through because they are very elaborate opinions. 2 opinions were published by the EGE in 2008. At this time, the EGE was able to publish 2 opinions in the same year. And these opinions, the first one that I would like to address is one called Ethical Aspects of Developments in Agriculture, opinion 24. And the second one, opinion 23, Ethical Aspects on Animal Cloning for Food Supply. Both address very similar questions regarding genome editing. The ethical developments in agriculture, of course, it applies to plants and it applies to animals. So any kind of source of food, agriculture has a lot of functions, a lot of ethics. So it's a very complex issue, but it's also our source of food, and it will be in the future anyway. And we are going to rely on plants and animals. And the third opinion is an opinion that we publish in the ethics committee of INRA, INRA Sirati Fremer, that I am still a member, and we published that in 2017. So what the first opinion, the opinion on ethical developments on agriculture said, I mean, it tried to develop about which are the priorities that new technologies should apply. This is the opinion, and I am reading. The AG's ethical approach to agriculture. The group considers the goal of food security food safety and sustainability are the first priorities of any new technology. Food security, provide an access to food to people, and we know that we have not reached this goal in the world with the crisis. This goal is even going backwards in Europe, for instance. At this moment, people are queuing in some places to have food in our cities. So food security is an important goal. Food safety, of course, it is essential, because without food safety, our food will produce problems that should never be produced. And of course, sustainability, because we have to ensure that future generations have an access as plenty as we are having at this moment. So these priorities should be applied to plants, regarding what Julia has said, and also to farm animals. I am going to say that farm animals will continue to be source of food in the future. Has been in the past. Human species, it's a hunter as well as a collector. So it's still probably some species. Maybe we are going to eat less bovine, maybe we are going to eat less meat in some cases, but it will continue to be. So I think that those are essential priorities. Second one, what the opinion 23 said on animal cloning. Opinion 23, I mean, it is animal cloning. It was at some moment, it was a hype. I mean, that animal cloning will solve many problems of food productions. Finally, we are not going to have any hamburger coming from a clone beef. Probably people in Argentina are using clone horses to play polo, but I don't think that this is the major technological development. So what we said in this opinion, that the EGE recommends that people addressing new technologies in animals address animal welfare. This is an important issue that I am going to repeat, that we have to consider animal biodiversity and sustainability. This is a very important issue. And of course, the public perception and acceptance of people. So regarding this issue, what are we doing with animals within genome editing? I think that animals in genome editing, we are using that as models for medicine. We are having thousands of mice edited in any possible gene on many genes. So nobody seems to be very worried about that. Second, we are taking back some ideas about using animals, for instance, for transplantation. It has been shown, for instance, in genome editing. One of the important issues that I think that I will discuss afterwards is that you can modify many genes at the same time. And they did that in pigs. So they allowed to annihilate a number of retroviruses that were a concern for xenotransplantation. So the idea of xenotransplantation is coming back, thanks to these technologies. We are also using other for instance, we are modifying, editing what we call usable animals, and Barbara will elaborate on gene drives afterwards, and of course on food production, as I said. So food production is these aspects. So what the opinion of INRA has said about the genome editing of animals. The first thing is taking, for instance, it's summarizing what kind of applications are important for farm animals. Farm animals, for instance, there is a continuous effort of breeding animals, new races that can be useful for farmers. So what kind of traits they are interested in? They're interested in traits that increase the health of the animals. Animals, for instance, resistance to different diseases. There are also modifications to the conditions of people perform animals in the field. It has been a very well-known example of dehorning cows. There's a whole discussion about that. This is feasible, has been shown to be feasible. The first examples had a problem with some residual DNA in the genome. We can discuss that. But in some ways, dehorning can increase the ways that animals behave in the fields. And then modifications of production characters. For instance, quality, less lipids in the meat, increasing some quantities of meat. So there are a number of things. So in this opinion, which is also available, and you can consult by the internet, the committee recommends that public scientists do research in this system. If we don't have public scientists having the best possible knowledge on all these technologies, we cannot advise the society. So, of course, the committee recommends that when doing science, you take into account questions like animal welfare. This is a concern. It is an important aspect. For instance, what kind of modifications are you sure that with these kind of modifications, when you are introducing some characters that essentially modify the animal? So all these questions are, and also, of course, questions of sustainability, as we have said. So I think that these are the main questions. And just to finish, I would like to address, half of 1 minute, perfect, to finish as we have been submitted a number of questions, I want to just highlight 3 of these questions. One, how easy is genome editing? I mean, genome editing is as easy as making a transgenic. It's not as easy as that. You cannot do that in your, a genome, an editing of an animal, of a plant in your garage. You need technology. You need to transfer the DNA to the plant, to the animal. There are a lot of species which are very difficult to transform. So and then to make a new plant. With animals it's even worse. For instance, one of the animals that we use a lot more is chicken. We have fantastic genetics on chicken. Chicken at this moment is maybe the best source of animal protein that we have at this moment. We rely on that for food production, and we cannot edit chicken. It's very complicated. We can, but it's very, very complicated. So, for instance, it is not as easy. The hype of easy, it's maybe have a contra. Then, what is the novelty? The novelty, Julian has said, what is genome editing doing is just producing variability, a variability which is needed. And variability, genetic variability, is the basis of breeding, of farm and animal breeds. And then when you have this variability, you need to enter that in these programs of breeding. That may take 10 years, 25 years, a lot of time to have the appropriate variety. So it is new. I repeat, for me, one of the most important novelties is can you modify 100 genes at the same time. But even so, it's more complicated afterwards to enter that in your breeding programs. And finally, there is a question about whether we can, genome editing is a way to create new species, something like that. Personally, I think that there are other technologies. At this moment, for instance, you may have heard that people are producing yeast, artificial chromosomes which are synthesized one by one. For instance, there you have a completely new synthetic organism, not by genome editing, which you are just modifying a small part. But we can discuss that afterwards. Thank you very much.
Thank you very much, Pere, for this very nice overview. And so for the last 45 minutes, we have been discussing latest developments regarding genome editing concerning being used on plants, being used on animals. Anne, Eric, Julian, Pere referred to multiple reports released by ethics committees, institutions, opinions, such as IMRA opinions on the use of genome editing in animals, courts ruling. So many words have been written on genome editing, yet many issues remain unsolved. And one of them is probably regarding the terms themselves. And this is why the last speaker of today's session, Barbara Prainsack, will present us her presentation entitled On Words and Values, the Power of Genome Editing, so we can take a bit a step back regarding the way we discuss and refer to this technology.
Thank you very much, Marion, and a big thank you to all the speakers who've already touched upon some very important issues, and also thank you to everyone who's taking the time to listen to us and hopefully to engaging with us in another 10 minutes. I would like to draw attention to some of the things in which I think debates and discourses on genome editing encapsulate some of the issues that reflect wider societal developments. So they speak to some concerns and topics that come up even now in the corona crisis. And I think when it comes to the genome editing policy debate and ethical debate specifically, making these visible can help us to work towards better solutions. And now that I talk about solutions, I remember that I haven't even introduced myself. So I'm at the Department of Political Science in Vienna, and I have been a member of the European Group on Ethics for the last 3 years. So I'm also involved in the genome editing opinion that some colleagues have already mentioned. Okay, but coming back to solutions and how words and values are important for that. So one thing that is really striking, and you see that on almost every end of the debate and in opposing camps, is this emphasis on openness. So Pere already said I would talk a little bit about gene drives as well. So talking about gene drives, one of the people who's very prolific in the gene drives science field and also in the debate is Kevin Esfeld at MIT, and he said, and I'm reading here from an interview that he gave to Michael Spector at the New Yorker in 2017. He said, the only way to conduct an experiment that could wipe out an entire species from the earth, these are insects here that he's talking about, is with complete transparency. And then the quote continues, for both moral and practical reasons, gene drive is most likely to succeed if all the research is done openly. So, great, right? There's nothing wrong with openness, right? But I think it's problematic in the sense because openness has now taken on the role of being an antidote to all kinds of ailments and ills of society, and certainly of gene editing's ailments. And as Sabina Leonelli and I have pointed out in another place, the rhetoric of openness and transparency, as important as it is, sometimes obstructs our view to the more substantive issues that are at play, such as values, such as social justice and distributional justice. So openness and transparency are ancillary values. They're not goods in their own rights. Sometimes they're bad. Think of your window. If you draw the blinds, it's not necessarily because you do something criminal behind it, but because not being seen, not being open can also make us, it can affirm our dignity, our personhood, and so on. So openness is not going to solve anything in itself. Openness is also often largely symbolic. It's a little bit like a medieval gallantry where you cover over grave injustices between genders, between people, by just, you know, being very gallant. So, and this is quite striking, and Julian has mentioned it and others have mentioned it, quite striking in the genome editing debate. Some social scientists, some ethicists are addressing this explicitly, but very often it's implicit. So we talk about openness and transparency when what we should be talking about is to how to alleviate some of the causes of the problems that we want to solve with gene drives in the first place. How we can alleviate injustices. And this is not only about access to gene drives, which is an interesting social justice issue in its own right, but also we should not use, and many others have said this, we should not use technological solutions as technological approaches as a solution to problems that in their roots are social problems. And we see this also, again, every day now in the context of the COVID debate. So some of the issues as they are configured in the genome debate are also prevalent and salient in other discussions. Another thing is, and this is specific to gene drives, but in the genome editing domain, but not, but it's also symbolic for something that we struggle with in society, is the rapid pace of things, the feeling that we cannot do anything to keep up. This is true for pandemics. This is also true for gene drives. So gene drives, as you know, are tools that change the way in which certain genes and therefore the traits that they encode are inherited. So they drive, this is why they're called gene drives, they drive a trait into the population by biased reproduction. So some traits are privileged through modification so that they become dominant in the way that they that the population reproduces. So gene drives in that sense represent a unique ethical challenge, not only because of the rapid pace by which they can change large populations of species, but also because of because in the context of gene drives, the very tool that modifies the genome is being released. So what we're releasing here is not the end result, but it's the actual tool that we release. So this connection between releasing a tool, higher level of uncertainty than a finished product, and rapid change is really a specific challenge. And in many other forms of genome editing, as you know, the finished product is being released. So when we are faced with having to make decisions in the context of rapid change, or decisions that might be very difficult to undo, also because of the traceability problem that other colleagues have mentioned, and we collectively tend to throw up our hands in the air and say, well, there's nothing we can do. Also, sometimes when we're faced with complexity, we say, well, you know, there's nothing we can do. And I think there's a lot that we can do. One of this attitude that there's nothing we can do is represented by calls for a voluntary moratorium, for example. So why does a moratorium have to be voluntary? And, you know, we have the law, we have ethics as something that helps the law to reach its goals and so on. There's a lot that we can do, but sometimes not only by, excuse the pun, manipulating that very problem that we're dealing with, but sometimes by changing some of the more structural factors. And that is really, despite the emphasis on social justice in some literatures, that's missing in the genome editing debate. That's really the big white spot on the map. So the next thing, and this is my last point, is the value of deliberation. And many observers have called for public oversight or control of how genome editing is governed. Very important. It's a constitutional issue in the deep sense of the word, right? We're using the words society's unwritten contract for genome editing. We're talking about a tacit constitution. These are the words that are floating around when people call for more public oversight over genome editing. And I agree with them. But if we look at these debates more closely, in some of them, the public, quote unquote, plays a very ambivalent role as a collective voice of moral reason, as a safeguard of a particular understanding of humanness or a particular social order. The public is very welcome, also as a legitimizer of scientific progress. But as a user of genome editing, the public is seen as a danger, you know, the biohacker. So lastly, as my colleague Claire Maris at City University has argued in a by now famous article on synthetic biology in 2014, when the public becomes too critical of scientific progress, when it becomes the sand and not the oil in the engine of scientific progress, then it is seen as an obstruction to public benefit, not only an obstruction to science, but an obstruction to public benefit. And then in an ironic turn, the public is turned against itself. And in many deliberative structures, there's a tight and rigid script that members of the public need to follow in order not to be seen as either a dangerous maverick that uses science in a way that is harmful or wants to use science in the way that is harmful, or an enemy to public benefit by hindering the progress of science. And the latter phenomenon, that there's a rather narrow path upon which, as a member of the public, one needs to stay in order not to fall down off the cliff of sin on either side, is of course also characteristic of the current COVID pandemic. Within which, as a good citizen, you can be critical of governmental policy, but not too much. You can criticize governmental policies to contain a pandemic, but not to the extent that it undermines the commitments and interests of powerful actors. And I'm not condoning violent demonstrators in Germany, and I'm not condoning men who use masks and say, I can't breathe, and thereby making light of the incredible plight of Black Americans who are killed by the police, and making light of women and marginalized populations who are literally suffocating, who literally can't breathe. So, but I think we need to listen to those people who say they are suffocating, and maybe some of them are suffocating in our particular political economy, and maybe it's an indication of that. So my very final point, quickly. Marion said I would speak about the power of words. Here, when we speak of editing genomes, we could use other terms, and that's a point of critique that we in the HEE often hear. Why don't we talk about modification, manipulation? Why don't we use the honest words? Well, Alessandro Blasime, as a team of authors, among whom was our very own Anne Combone-Thompson, wrote an article about this in 2015. What's in a Name? Highly recommended. And what emerges is then when we talk about editing genes or genomes, we're doing two things. First, we are avoiding the use of other terms, such as manipulation or modification, that have different negative connotations. Editing, on the other hand, has a connotation of care, purpose, improvement, whereas manipulation would by most people be seen as bearing a negative connotation, also because of its use in common parlance to refer to people who try other people to get things that harm them. So, but the term editing is not really neutral either, although this is often the assumption that editing is the most neutral term. The term editing evokes the metaphor of writing, which could also be seen as highly misleading in this context. Dating back to the early days of the Human Genome Project, the international attempt to map the sequence of the human genome, as you know, and writers such as Kay, Nalkin, and Lindy have drawn attention to this. The genome was compared to the book of life, which, once the language had been decoded, could be read like one reads a text. And the tenacity of this genes of information metaphor, and the fact that it has become the dominant metaphor of the domain of gene editing, can partly be explained by the deep way in which it had been inscribed into public and policy discourses about genomes. Leaving technical expert jargon aside, it's very difficult to talk about DNA outside of the metaphor of a text. And given that this information metaphor has led to misleading expectations in connection with DNA testing and personalized medicine, it could be argued we should stop using it, right? But how should we call it then? We could also argue, though, that the metaphor of the genome as a text could be employed, but with critical scrutiny, and to make us ask productive questions. If we're editing genomes, who has the editorial rights? Who wrote the text? And what does the text mean? Who should agree on what's written? And can different versions of the text be erased, right? Is there a correct original version of the text? So I conclude by saying that if we ask these questions, it's not just an academic cultural exercise, but it actually helps to better understand what we are afraid of and why, and what we should hope and why.
Thank you very much, Barbara. So we see very well with this panel that debates on genome editing, whether we address them from a scientific angle, an ethical one, a political angle, etc., are particularly complex. And Barbara said openness and transparency are not solutions to themselves. And I think it resonates interestingly with the motto of this 2020 edition of ESOF, which is freedom for science, science for freedom, as Anne recalled us when we did our rehearsal session. So it is not because science can than science should. So I leave the floor to Dominique Leglu, who will now take the lead with additional questions to our speakers and interacting with the audience. Thank you.
Thank you, everybody. Hope you can hear me. I was very pleased to hear you all, especially Barbara talking about editing. I'm the editor-in-chief of 2 magazines in France, Sciences et Avenir and La Recherche. And of course, we know that the editor is not neutral. Apart from that, of course, you said so many things that it's not easy to start with a single question, but I will try. Somebody mentioned, of course, that we're in a time of COVID. I thank you, all the people who watch us on Zoom or elsewhere. Maybe some are in Trieste. Hello, Trieste. And my question would be that COVID has shown us that with the social networks, not only them, a lot of fake news and conspiracy theories were evoked, started. And you all remember one of them concerning this lab in Wuhan, and saying that the virus itself was cloned or whatever, was edited or whatever, manipulated there. And of course, many scientists has to show, to make clear that it was fake news, but you still have people going on with that. When you will use the wording gene editing, even for animals or plants, where will you say that in a time of social networking, social networks? Where do you do that? And I refine my question. Maybe Eric might like to answer first. When he said that, well, we have conventions, but what do we do with them? There are deliberations in conventions, so it doesn't work at all anymore. Where can we have a good, serious deliberation, and how to present it to the general public, which is so different from one country to another? Eric, maybe you say it's impossible. Maybe you say, as Barbara said, no, it's always possible. We have to work. We have to go on.
Well, thank you for the question. I will say, to borrow the genomic analogy, I have an optimism gene. It is it's quite penetrant in me and highly expressive. I don't think you can work in a field where there is so much technology, science, ethics, and policy moving, and not have some hope for the future that is positive, because the alternative is just too depressing, quite frankly. So maybe a couple of quick ways of responding, and I really appreciated Barbara's presentation. This isn't to push back on her point, because I actually agree with it, but just to be a little provocative. I do think there's a time and a place for when a particular word or phrase captures the attention of the public and the media, and there is absolutely nothing you can do to reverse that and extract it from conversation. It's just impossible to do that. Look at the debate that happened for many, many weeks at WHO to push back on what the name of the virus would be, what it would be called. Many, many pages, many tweets, many discussions. This is not unique to COVID. We saw the debate over the naming of the HIV virus, Montagnier and Gallo. This is not a new topic. What do we name or call things? I do think that it gets a little tiring and exhausting to fight about words if they don't matter. If we're going to call it gene editing because that's what's happened, and it will take more energy to not call it that, then let's move on and solve the problem. But note, I think, as Barbara was saying, that words do matter. That's point number one. I'll make my second one because I think it's really where you were. And by the way, we saw this with cloning, right? We saw this in, I was still just coming onto the U.S. Bioethics Commission in the middle 1990s after Dolly was cloned. And, the attempt to call this technology somatic cell nuclear transfer was just a non-starter. It was cloning. I mean, that's what it was. Move on. So there is a bit of pragmatism on the policy front. Now, how does that translate, as I think you asked, into the public square? Well, it may also be, and this is a different gene I have, which is to be a little bit mischievous. It's very close to the optimism gene, by the way. And that is, we should probably suspend the idea of having one place where all of this happens. So one definitive environment, sort of the original document, so to speak, the original environment. Will it be a citizen assembly? Will it be a legislature? Will it be the science community? The answer is yes, and which is what makes it all the more challenging. How do we harmonize, if that is even another word we often use in public policy, attempt to find a harmonized document? We can go down a musical line if we wish and say harmony is many different notes, not just one. So I think my observation to come to a close is that the way that the wording affects public discourse is an artifact of the speed of the science, technology, and ethics. I don't think anyone cares that Dolly was a cloned sheep. They just sort of remember this technology. It's like IVF, it's like gene therapy. We shouldn't have called it gene therapy when it first started. It was gene transfer, but we couldn't get people to change their minds. So I think we should bracket the wording, remember what it stood for when we initiated it, and now think about how we can translate the discussion into the various publics, plural, not simply pick one translation for one environment and hope that everyone else gets it.
So think universal and word it local. Anybody wants to answer? Also, Anne, would you have something to add to what Eric said?
Sorry, I had to have my microphone. I think this is very true, and what Eric said, and very realistic, I would say, which is important. And joining what Barbara said, words matters. And I think that the important thing is not so much to use one word or another one or a third one, is to reflect on what the word we use mean or call as images, as reactions, and so on. And this, I think you can do with any public. Reflect on the word you are using and giving different perspective on that. And I think that starting with the words can be a very good way of starting a debate, because the words, whether they are different or are a tool that everybody in the human species are using. And I think this is a good way, starting with words, to start a debate, which is in the end the important thing that must happen when people must have a dialogue. That's it.
Before even passing the word to Barbara, if she wants to react, I would like to remember to the audience that they can ask questions and send them, and I would be happy to reflect them to the right participant and the right panelist. Barbara wanted to react to Anne and Eric, or nothing to add, and we pass on to another question.
We can pass on. I think it was very helpful what both Anne and Eric said, and of course I don't think that talking about words is the most important thing, but as Anne was saying, I think it can be useful to take it as a starting point, actually, and it's definitely important to reflect on it. Yeah, no, but I have nothing to add beyond that. Thank you.
I would have Julian asked to answer, but I had a question for you, Julian, because I want to understand better. You mentioned, and that's an important thing, that in Europe now gene editing things are the same thing as a GMO. Is there a possibility that there is a step back and that there's a new discussion and then it's not like a GMO? Do you think there's a possibility of that? So it could make a real change. And well, you emphasized what complication it could lead to if we want to have traceability and all these sort of things. So what do you think? What is the next step? Is there a step that you can foresee in 2 months or 1 year of something important where all this will be discussed very officially?
Thank you very much, Dominique. A number of things. First of all, on the words, I just want to indicate that the way we think in different languages is really important. For example, in French, Spanish, Italian, the word biosafety and the word biosecurity are all the equivalent of biosecurité. So when we talk about words, their understanding in different languages is very significant. And that was the first point that I wanted to raise there. Does the European Commission intend to think about changing the directives so that we can address the simplest of modifications using gene editing because of the precision? And the answer is that they are thinking about it. We can't completely say we don't need regulation for gene editing, because we could actually change, for example, the range of light which plants use for photosynthesis. And that would be a major change, enough to cause us to think quite hard. But to change one base, for example, I use a human example, to change the base in the beta chain of hemoglobin from one amino acid to another, it's just a single base change, could solve the problem for hemoglobin S, sickle cell disease, and hemoglobin C, another disease of hemoglobin which people suffer all over the world. So we've got to be aware that there are different ranges for what we're thinking about. So are we thinking of changing? Yes. Should we be? Yes. Should the action that we take be proportional to the risk? Yes. And I'd go further. I'd probably argue that we shouldn't say gene modification needs regulation, but modifying using chemicals does not. That seems to be wrong to me, because the same arguments apply. So should we be applying the same arguments? And should we be thinking of agriculture as a holistic concept, all the way we use the land? And that becomes important when we think about climate change, because the kinds of plants we're going to be able to use in particular environments are going to change. The amount of water we need is going to change. The amount of water that, say, South Africa, which I live in for much of the year, needs is already limited, and we're already restricted to relatively small amounts of water in much of South Africa. That includes farms. How do we produce enough food if we don't have water? By using drought-tolerant plants? How do we produce those? So these are the sorts of things we have to think about. and decide whether or not we're going to ensure that we can produce the food, feed, fiber, and fuel that we need.
Thank you. Pere, I wondered whether you wanted to react. Yes, do.
Just a quick answer. One regarding wars. I think this is a very pertinent discussion. Just 2 points. One, of course, at the beginning of the discussion about genetically modified plants, the word Frankenfood was used. No? So, well, people are using words in many different directions, in many different ways. And we have to think about how they are using. Second thing, I will also refer to an opinion of the NAFIL Council of Bioethics about the word natural. I think it's a very interesting report. And I think finally the conclusion is don't use natural. Or if you use natural, explain what do you mean by natural. There is another word that I think for me it's very dangerous, which is normal. So I mean, you're right that these words, but there are around very interesting discussions about that. And just referring to your question, Dominique, the point is France. France is terrible. The position of the French government and the French society, it's very surprising. I understand that the problems of agriculture in France are very complex, and that France is the most important country in Europe for agriculture and for food. But I don't understand why in France it's so difficult to put some small sense. Now you are going to make any mutated plant forbidden. This is foolish. So completely, and people are talking about that very easily. So in France, we're talking about different countries. I think the position of France, which is very important, it is central one. It should be Cartesian. You see, you should think about the card. It's one of the most difficult.
Thank you for these remarks. I won't see myself as the representative of the whole population of France and of our government, but remain a journalist who asks questions. And I noticed one of you, I think it's you, Perry, or Eric, sorry, I can't remember, who used this wording, unnatural selection. You use this? No. But of course, this is the kind of thing which is striking and that people remember. Now, we have some minutes left. And of course, I think for the audience, even if we've been talking mainly about plants and animals, of course, we want to understand, and I would like to know very simply, what do you think will happen next? I heard Julian say, if I change just one base, I can eliminate one disease. At one point, won't population ask for a change if a little fetus has a disease and you don't want to abort, but you want to have a normal child? And people will continue to say normal, the usual population. So what do you think will happen? If I may say so, as a journalist, I frankly think that at some point, yes, the box will be open and there's a danger there. But I think we'll find, some scientists find, some medical doctors will say, I can cure you with that gene editing system. So what do you think about that? Julian wants to answer, but I would like to have all your feeling also to know where we're heading to. Thank you, Julian. Go ahead, open your microphone.
It's funny, the different reactions to different things. People haven't complained in general about GM for vaccines. They complain about vaccines, but not GM. They complain about GM for food. They haven't objected to medicines produced by GM, and they haven't complained about things like vitamin C added to food, which is made through GM. So we have different attitudes in different spheres of activity. Now, would the public say yes to using these techniques in medicine? I think the answer is unequivocally yes. Would they say yes to it in food? In many countries in Europe, especially France or Greece, they will say no. In Britain, they will say yes. In America, they won't know. Apologies to you, Eric. So these are the differences, but in medicine, yes, unequivocally. And that's really rather interesting. You can do it to my body, but you can't do it to my food.
Eric, would you have anything to say about that?
First, I'm speaking to you from Ottawa in Canada, but your apology is accepted since I hold citizenship in both countries. I think you've actually, Dominique, you raised a point when you introduced this that I don't want to let you off the hook on when you were posing this as, is this all fake news? A term that entered the lexicon, as far as I can tell, about 4 years ago with the election of the guy south of the border, and has now taken on like wildfire. It's used as an accepted phrase when in fact in science, in philosophy, in political science, in law, areas where, as we were saying before, the use of words matters, the specificity, the stipulation, this is what I mean when I'm using this word, that those rules are gone. And they're gone for reasons that would take an entirely different panel. So I make that point. I would say that, again, to be a little bit proactive, I think provocative, I think the box is already open. It's not a question of whether it's going to be open. Barbara alluded to biohacking and DIY, synthetic biology, and I mean, it's there. In fact, the issue is not whether, it's who's doing it that we don't know about, and are they simply offshore sort of rogue hacker types who do it for the kind of bizarre curiosity, or is there something more nefarious going on? So I would agree with you, though, Dominique, that I think the idea of whether these techniques, the collection of techniques using the CRISPR platform or others, will be used sooner than later, I think the answer is yes, for probably two reasons. One is, quite interestingly, and unlike the recombinant DNA debates of the early 1970s, this technology came out with sort of two arguments in parallel. One was, this is an unbelievably simple, Perry's point notwithstanding, this is an unbelievably simple technology. Lots of people can do it. Maybe not in their garage today, but it was framed as, this is easier. I mean, it's sort of a revelatory statement. Whether that's true or not is beside the point, but it came out that way. People didn't say that about gene therapy. This is pretty simple, anyone can do it. This was a thing that was accessible. And secondly, it kind of sneakily brushed aside many of the fundamental ethics issues that have occupied in vitro fertilization, stem cell science, and even the recombinant DNA work. So this was, you don't need embryos, you don't need fetal tissue. So the sort of big ticket moral objections were at least bracketed. They may not have been completely dismissed, but they were bracketed. Once those two arguments, the horse had left the barn, it's only a matter of time. And now we're just trying to figure out which will be the, I wouldn't call it the winner, but what will happen first. It might be thalassemia, it might be sickle cell, it might be one of the rare diseases. So to me, it's not whether, it's which one.
Okay, very clear. If somebody wants to react, we've got little time. So Pere, and maybe Anne, you will conclude. Okay?
Okay.
Let me just repeat what I said. I mean, technology is not as easy as that. You need to be well prepared. Second, there are still some collateral effects that are very easy to control in plants, for instance, that are relatively easy to control in animals, that are very difficult to accept in humans. So, and third point, what you are going to modify? What genes are you going to modify? I think that our knowledge of interesting things is not as big as that. It's increasing rapidly. It's increasing, but it's not as that. So candidate genes are not famous. And for instance, many of the single gene diseases are being solved by pre-diagnostic, by diagnosis in the fetus. I mean, and they are doing. I mean, some of these diseases are rapidly disappearing now because people are using diagnostic, pre-implantation, proper birth. So we are doing that at this moment without any need of gene editing and whatever it is. So there are very rare cases where the question will be solved by editing, but it will be very rare. So I think that as in plants, the first demonstration was in '83. The first plant was planted in '94. Now we have hundreds of millions of hectares every year planted in the world. In plants, it will happen because it's so evident that it will help farmers, that it will happen. And then some seeds will enter our harbors in Rotterdam or whatever, and we will be unable to say, but maybe technologies in Europe will be less influent that it has been in the past.
Anne?
Yes, thank you. So I will react on different things that have been discussed in the panel. And one is, you know, I'm French and I'm not a moderator, so I can talk about some of the reaction in France without being a representative of French society. I think that for food and for health, what you said, Julian, is very true. And for health, people say yes, even to a certain risk, when there is no other way to achieve health. And in the cases where we could envisage the use of gene editing and already the use in clinical trials with the somatic cells, it's used. And this is one way where no other way is better or seems to be better. Whether for food, and especially in France, for eating well, and in many cases having enough to eat, why in whatsoever gene editing would you need? So I think in the population within one country and with a number of traditions, it's the use of technology is more accepted when there is no way to achieve what is the good you want to achieve. And I think this is part of the discussion, and only part, of course, of the attitude in France regarding food. Regarding the disease, and yes, I agree with Eric. It will happen at a time. The question is, who is going to decide, advise on when is the good time and for what? And I think the discussion, we could have a second session on that, yes, is, we are going to finish that, is the conditions in which you could say, okay, let's go one step forward. And I think with this, and step forward is a nice way of concluding a session. And I would like to thank all of you, because this session has happened because you said yes to this idea of having it proposed, and yes to this virtual adventure that we are all living with. And so thanks to all.
Thank you to everybody. Thank you.